Replication of viruses in HIV, attachment and absorption. Step 1. The virus binds to the host cell in a process called attachment. Step 2. CD4 molecules present on T cells and macrophages can be recognized by HIV proteins. Step 3. The virus is circular in shape with spikes of a protein called GP120 around its circumference. As the virus nears the host cell, GP120 extends from the viral envelope and connects with the CD4 receptor. Step 4. Next, the virus binds to co-receptors CCR5 and CXCR4. Once the virus is bound to the receptors, GP41 activates. Next, the lipids connect, the virus delivers the nuclear capsid of HIV into the cell cytoplasm, and the ENV gene helps with this process.
Entering and decoding, step 1. Once the nucleocapsid is delivered to the cell cytoplasm, the process of encoding begins. Step 2. Cellular enzymes strip away the capsid shell, and the genome is released. The genome contains 2 viral RNA strands and 3 essential replication enzymes, reverse transcriptase, integrase, and protease. Step
Reverse transcription biosynthesis. Step 1. Each virus has 2 copies of its RNA genome, and each one is attached to a reverse transcriptase. Reverse transcription. The reverse transcriptase enzyme transforms a single strand of RNA into the double helix form of DNA with the help of the PLL gene.
DNA then moves towards the nucleus of the cell using the VPR gene.
Integration of provirus. Step 1. The process of integration incorporates viral DNA into the host cell's DNA strand using a viral enzyme called integrase and the PLL gene.
As so. Stage 2, provirus. At this point, DNA molecule is considered a provirus. Replication. Step 1. Activation of the cell induces transcription of proviral DNA into messenger RNA. The infected host cell is replicating the virus without knowledge of its existence. Step 2. Viral mRNA is then synthesized into building blocks for a new virus.
Production of main virus. Some of the proteins have to be processed by the enzyme protease. Protease cleaves the proteins into capsids. This is the original viral protein. Step 2. The different components of the new virus begin to assemble at the edge of the cell, like so. And the core proteins assemble around 2 RNA strands and the 3 main enzymes, creating a new capsid that can travel out of the host cell with the help of the GAG gene.
Step 3. This new capsid leaves the cell, enclosing itself. Budding. HIV capsid leaves the host cell using the ENV gene. The virus matures and becomes ready to infect other healthy